Great, thank you. Good to be here at Q2B again. I'm Paul Lipman, Chief Revenue Officer at Infleqtion. I'm joined by my colleague Peter Noell and by Ian Njoroge from CSL. So, gentlemen, why don't you introduce yourselves?
Yeah, sure. My name is Ian Njoroge. I'm a senior scientist at CSL. Been there for several years now. Have a interest in quantum, but typically it's just something that we've been exploring in the last couple of years. So.
Yep, and I'm Peter Noell. I'm a solutions manager with our quantum software group in Chicago. Primarily focus on business development, but also work closely with our quantum software applications team. So we've done some prior work in life sciences and healthcare through programs like DARPA Impact and the Welcome Leap Quantum for Bio program, which is still ongoing. We're in phase 2 of that. So connecting with CSL was a great opportunity for us to transition some of the solutions that we've developed through programs like that and some tangential work there into some industry relevant problems working with a great industry partner like CSL Behring.
Great. So Ian, first question is for you, and I know we have some slides, so I'll maybe ask the question first and then bring up the slide. Maybe we can start, tell us a bit about CSL. What does the company do? A little bit about the topic is modeling protein chromatography. So a bit about what is protein chromatography, why is that important for CSL, and maybe more broadly, why is it important for the world at large?
Yeah, so CSL is a global biotechnology company driven by our promise to save lives. We deliver life-saving products to patients in over 100 countries, generating over $13 billion in revenue. In the downstream manufacturing of biopharmaceuticals, chromatography is a key process that enables the high selectivity separation of proteins. The global process chromatography market size in 2022 was $12 billion, and it's projected to grow to over $27 billion by 2032. And there are different modes of chromatography. There's affinity chromatography, ion exchange chromatography, hydrophobic interaction chromatography, and multimodal, which kind of combines some of the different modes together. Typically, you can run it in either historically a batch process or, as is recently coming to or being innovated on, as a continuous process, as well as in either a bind-in-the-loop mode or in a flow-through mode. And so here, there's a graphic on the left side of the screen that kind of shows an example of a chromatography process. So typically, you'll have a column of resin, which is typically a porous bead that may or may not be functionalized with a ligand, depending on the interactions you're looking to have with your protein of interest. You'll typically load your sample for separation on the column, and then introduce a mobile phase to enable transport of that sample through the column. In affinity chromatography, for example, you're relying on a capture of your protein of interest, and then introducing a second mobile phase that will then interrupt that interaction and enable you to capture your protein of interest. But the mode kind of shown here is sort of in a flow-through mode, where as that mobile phase is carrying the proteins through the column, you end up getting interactions with your column material, resulting in a separation of your protein, and then you essentially capture the fraction of interest that you're interested in.
Great. So Ian, I know we worked with CSL. Peter specifically worked with CSL to use quantum computing to model this protein chromatography process. What were your objectives? What kind of problems were you trying to solve? What were you trying to discover in doing this work?
So in chromatography modeling, there are typically two main approaches. One is empirical, where you gather a large amount of data about your process and use statistical methods to essentially build a model of your process. However, due to process complexity, these usually have limited utility and are also not able to be scaled up as you are building your process from a lab scale to a manufacturing scale. The more preferred methodology of modeling is a mechanistic modeling, where you take a mathematical model based on causal mechanisms. And these typically tend to be multi-scale. So you're looking at processes at the column level as well as processes at the particle level. At the column level, you're mainly looking at transport processes in terms of convective flow, so the velocity of your mobile phase through the column, as well as axial dispersion, so diffusion of your molecule across the column as it's flowing through the column. At the particle level, you also have transport processes in terms of the molecule transporting into and out of the porous material of your stationary phase, as well as absorption kinetics, especially in the case where you have a functionalized bead where you're looking for specific interactions with your protein of interest. Typically, when this is modeled today, there's an abstraction of those absorption kinetics and equilibrium, where a general isotherm is basically used for that, which leads to several limitations. The first of which is nonlinearities that do arise when you're looking at very high loadings of your column. And typically, for efficient separations, you do need these high loadings. The second of which is there are typically no analytical solutions to some of these isotherms, so you end up relying on numerical solutions. And there are several software packages that will do these types of modeling for you today. However, these limitations and not having that molecular level interaction modeling results in a lot of trial and error experimentation in order to fit your model to the specific use case that you're looking to do. And so a mechanistic model that's able to incorporate these molecular level interactions would provide a lot of value. And as a first step to that, we looked at essentially generating a model of the packed column. And that's where we leaned on our collaboration with Infleqtion to look and see how quantum could provide an advantage in modeling those packed columns.
Great. So, Peter, next question is for you. I mean, this seems like a very complex, sophisticated problem to address. Why did we select quantum computing to try to advance the kind of state of modeling this process, and how did we go about doing that? Maybe you can kind of take us through some of the details there.
Yeah, absolutely. If you could click to just one more here. Yep. Thank you. So I'll talk a bit about how we decided to model this chromatography process, and I think as I go, it'll become clear why we thought this would be a good fit for quantum computing. So backing up, this is a sphere or particle packing problem, and there are a few different ways and variations, different flavors of the problem. So one consideration is the problem bounded or unbounded. If it's unbounded, then it becomes a lot easier. You can think about packing together soccer balls together on a field. In this case, we're working on the bounded instance because it's in that cylindrical column. That's where the chromatography takes place. And if instead you are packing soccer balls in a bin in your garage, the problem becomes a lot more complicated because of the boundaries. So boundedness is one consideration. Another is two-dimensionality versus three-dimensionality. So we looked at cases of both, circles versus spheres. And then finally, whether the radii of the spheres and circles that we're trying to pack are homogeneous or heterogeneous. So again, we looked at cases of both. So starting in order of what we did from simplest to most complicated, first you can see on the right there, we started with the two-dimensional homogeneous circle packing problem. So what we did here is we mapped this as a graph problem and solved it with QAOA using our SuperStack platform on the IQM Garnet device. The reason we used IQM was because we, as Pranav Gokhale, my colleague, mentioned in his talk yesterday, in our software group here, we like to work with a wide variety of qubit modalities, and our quantum computer scale was previously being used for our work with NVIDIA that we announced yesterday. So we used the IQM device because of its relatively high fidelities and its qubit topology that somewhat resembles a lattice. So the way we solve this is we superimpose this qubit lattice on top of our circle, which is the two-dimensional version of that protein filtration column. And what each qubit represents is a node where we may or may not center one of our circles as part of our solution. So if you see on the top right of that plot there, the red dots mean we ended up centering a circle there as part of our solution, black meaning not. So we're using the quantum property of superposition here to determine the 0 or 1 sort of aspect of that, whether or not to make that decision. And then you'll also see the edges of the plot represent whether or not two circles centered at those points would overlap. That means that we can't center circles at both. So it boils down to a maximum independent set problem, and that second portion of the edges uses the property of entanglement. And we solved this to optimality on this 20-qubit device, and we're very happy with those results. We also noticed that if you have more qubits, you can more densely discretize the space there. So you basically have a wider selection of places where you can place those points. And in some cases, that will lead to better results, but at the cost of the overhead of more qubits. So that was the first case that we produced results on. The next case is the middle one there, the heterogeneous circle packing results. And those results are from classical simulations. So this is modeled exactly the same way, except at each of those candidate nodes, there are log n qubits needed to represent the n different radii. So you have blue circles, yellow circles, red circles. You need more qubit overhead to model that. And that's currently intractable on devices like the one we chose here, just because it requires a bit more qubits, even though it scales relatively favorably. And then finally, we looked at the heterogeneous sphere packing problem. So in 3 dimensions now, this is really the holy grail here, and the model that most closely resembles what's happening when we're looking at CSL's protein chromatography problem. And this, very quickly, we found becomes intractable for classical computers to be modeled this way. And while we still can't solve it today on quantum computers, there is some positive outlook on what we can do there in the coming years.
So, Peter, we talk a lot at Q2B and in the industry generally about the time frame for quantum advantage, but perhaps kind of think in terms of commercial advantage. What is the outlook here for commercial advantage for protein chromatography in your opinion, and when do you expect we'll actually see these kinds of solutions deployed in production?
Yeah, I think we could see some quantum advantage here in the next 3 years or so, given the roadmaps of Infleqtion and others in the industry to scale to thousands and tens of thousands, even hundreds of thousands of physical qubits here. That would, even if they are still noisy physical qubits, you can model a much larger problem here than what's possible today classically. So you could use an algorithm on those physical qubits like we're using today, QAOA. And I imagine that some more interesting logical, an analogous logical algorithm to QAOA will emerge to run this as a logical circuit as well.
Got it. So maybe, Ian, a question for you. So Peter's saying, hey, look, in 2 to 3 years, the quantum computers will have advanced to the point where we can actually solve the 3D heterogeneous sphere packing problem. Let's say we're back here in Q2B in 3 to 5 years' time. What do you think the economic impact is for the biopharmaceutical industry, specifically for protein chromatography? Is there a way to perhaps put a number on that?
Yeah, I'd say so. Globally, the protein development or chromatography development is about a $9 billion type market. And so with solutions like this, you can probably envision anywhere from a 20 to 80% improvement in time in terms of developing that. And so that, scales to anywhere from, $2 to $7 billion in terms of resources that you can either deploy elsewhere or, return to you in terms of time saved, in terms of resources saved. And so whilst, the modeling here in terms of 3D, there'll still be requirements in terms of adding functionality to really enable that molecular level interaction modeling. It's a very large gain that you can get if you're able to enable that here in the next near-term future as well as in the long-term future.
That's fascinating. That's a very significant potential impact, not just from a financial standpoint, but in terms of protein chromatography, if I'm understanding you correctly, has such a huge impact on quality of life for probably hundreds of millions or billions of people around the world.
True. Yes, that is accurate. In terms of what you'd be able to see from a time to market perspective, you'd be able to decrease that significantly, and also in terms of being able to scale up from a lab scale process to a full operational manufacturing process would reduce the amount of time that it would take for therapeutics to hit the market. So yes, you're correct in terms of there's also a societal benefit in terms of being able to develop new drugs and drugs that are able to address concerns that we may not be able to do that today.
Fantastic. Yeah, it's really, I think, very encouraging to see the impact of quantum in the life sciences area, because it affects all of us in such a personal way. Maybe one last question. I'll open it up to both of you. So we've talked about a very specific application of this sphere packing problem and the use of quantum computing to help to address it. Do you see ways in which this kind of methodology, this algorithm and approach has applications perhaps in other industries or for other problem sets within the biopharma arena?
I would say so. Right. If you're able to pack, or at least physically represent molecules and also add chemical functionality to them, that enables you to solve a wide ranging of problems, not just in terms of chromatography, but other separation techniques that we utilize. You could also enable you to do bioreactor type modeling as well, which is useful in that, we discussed downstream, which is usually the separation part to get to your quality molecule, but the upstream where you're actually creating that molecule either in a monoclonal, in a cell or in cell culture or in other modalities, that can be very useful. So being able to model the entire chain is very useful, because you then are able to also optimize your process from start to end, enabling you to be more efficient, be more productive, which could result in a lower cost of medicines in the future as well.
Yeah, I'll add to that. Even beyond just the purview of biopharmaceuticals, this work builds upon some other work that we've done in other industries. Like, for example, the work we've done with financial services companies like Morningstar closely resembles the underlying math of this problem. And like in this example you see here, that it could be very similar to portfolio optimization. So you want to maximize return and minimize risk. It's sort of the superposition and entanglement that we see there. So the ability to solve those at large scale on the order of thousands and tens of thousands of qubits is really, really exciting.
Very good. Well, thank you both very much. This was absolutely fascinating. I think we have a minute left for any questions. I'll advance to the Slido if there's questions. I know we're one of the last sessions of the day, so we'll be more than happy to take any questions from the audience here.
Thank you very much. If you have questions, please submit them through the QR code. I don't see any questions right now, but
great.
Maybe
Thank you very much. And if you do have questions afterwards, certainly feel free to reach out to any of us here. We'd be more than happy to discuss with you. Thank you. Thanks very much.
Thank you, Paul.
Thank you. Thanks, Paul. Thank you.